How you doing?
Oh, hey. Hey, Robert. I'm having trouble unzipping my jeans.
But you're not wearing jeans.
Well, not my blue jeans, my genetic jeans. The zipper to my genetic information isn't working properly.
Okay, you mean like Helicase?
Yeah, exactly. The enzyme responsible for separating the DNA double helix during genetic repair and replication.
DNA helicase is commonly a circular enzyme that surrounds and catalyzes the unwinding of double-stranded nucleic acid chains, such as DNA duplexes or DNA-RNA hybrids. Helicase is always present within the cell nucleus of eukaryotic organisms to allow continual modification and repair of DNA. Because prokaryotes lack membrane-bound organelles in a nucleus, DNA helicase floats around in the cytoplasm, concentrated around the nucleoid, which contains the genetic information. The expression and concentration of DNA helicase increases dramatically through the S phase of the cell cycle and speeds up the rate of transcription. Now let's take a closer look at the structure, function, and mechanism of DNA helicase. On to the structure. Like all enzymes, DNA helicase structure varies from organism to organism, but DNA helicases are usually in a hexameric ring which surrounds a single strand of DNA. Within a single organism and even a single cell, multiple variations of the helicase exist. These different variations between helicases probably relate to their function. For example, some helicases may include an additional domain to recognize the methylated portion of DNA, which indicate mutated, damaged, or mismatched base pairs, while others, without the additional domain, separate DNA during normal transcription. Now onto the function and importance. DNA helicase separates two strands of DNA during transcription and replication. It also separates a DNA-RNA complex during translation to allow DNA polymerase to place the corresponding nucleotides in order to replicate the DNA strand. This replication is necessary for mitosis or for protein synthesis. It is also critical for the repair and synthesis of new DNA, permitting the duplication of genetic information, which is necessary for sustaining life.
Mechanisms of DNA helicase. Motor segments within the DNA helicase use the energy from hydrolysis of ATP as a means to unwind energetically stable duplexes of DNA. Helicase accomplishes these functions by first oligomerizing its subunits around a DNA double helix and surrounding both strands. The quaternary structure is stabilized by the binding of either NTP or a metal ion to the exterior of the enzyme DNA complex, depending on the organism. 7 motifs are conserved between most families of helicase, which play a crucial role in binding ATP as well as DNA. The residues involved include phenylalanine 183, aspartic acid 110, histidine 85, asparagine 352, tryptophan 250, histidine 580, tyrosine 248, glutamic acid 215, threonine 29, lysine 28, arginine 278, and tyrosine 270. The proximity of the ATP and DNA binding sites is crucial in forming a transient covalent bond to the sugar phosphate backbone. The hydrolysis of ATP provides the energy necessary to overcome the activation barrier binding the enzyme to the DNA backbone. The 3' portion binds to a negatively charged groove, while the 5' end binds to a cavity formed by a couple of subdomains. Consequently, the formation of a transient covalent bond on a single strand of DNA, combined with the size of DNA helicase, places steric stress on the hydrogen bonds between adjacent nucleotides of.
Just in case you haven't noticed, DNA helicase is essentially a genetic zipper, wherein the tape and teeth are respectively analogous to the phosphate deoxyribose backbone and nucleosides, while the pull and slide are akin to ATP and helicase. Fortunately, we have a special guest to help demonstrate how helicase works.
You're close. Give them to me.
Trust me. Hasta la vista, baby.